Dropbox.
How's everybody doing today?
Oh, right.
All right. I just now put up some updated slides. There's nothing really all that strikingly new about the slides over the presentation that was posted last time we met. I've just added the Hannafin et al. Slides to the Yapney presentation, but none of the Hannafin et al. Slides are slides that you don't already have access to you in the paper itself. So any questions before we get started in terms of what we were covering last time? Dr. Judd, do you have any announcements or anything?
I don't. I'm getting different evaluations from folks in lab, which I asked for by today. So I'm glad to see that coming in, but I don't have any specific announcements yet.
Okay. You'll notice that the gradebook is being more and more populated as time goes on. I spent like the last 3 hours cleaning up the gradebook somewhat. So as you look at the gradebook, recognize that the homeworks, they may total to more than 75 points, or they may total to less than 75 points. In the end, the way that homeworks count is whatever homework that we have, and the point total varies from year to year. We look at the proportion of the homework total that you earned, and then we scale that up or down to 75 points. So as you look at the grade book, recognize that Moodle doesn't handle those kinds of machinations very well. As a matter of fact, when it comes time to report your final grade, Dr. Judd and I download the scores that we have in Moodle, and then we do all of the calculations ourselves, because Moodle doesn't do a great job of adding up all your homeworks, dividing by the total number of points that your homeworks were worth, and then scaling that to 75 points, which is what your homework grade is. So just that caveat as you continue to look at the grade book. There were some things that only half of the grades were in, because either Dr. Judd or myself hadn't put in their half of the grades. I think I've cleaned up a lot of that so far. The Hendry et al. Was only had grades that I had graded, and I think it's because my computer was filling up and I took all the graded assignments off of the Dropbox folder before Dr. Judd could get them entered. And so I entered all of his grades earlier today as well. So the grade book is in better shape than it was 3 hours ago. So that's clearing up and getting towards the point of being caught up.
As I told, I think 2 of you in emails today, I'll be working on updates this afternoon on the same thing.
Yeah, I got out of class this morning at 10, and I've basically been doing nothing but grade book oriented organization and cleaning up since then. So, all right, then where were we last time? So we were talking about the Gaffney paper. Let me pull up my presentation. And where did we end up at? Well, we were talking about chimeric DNA, weren't we?
Yeah, we asked everyone to look up what a chimera is and why xenopus eggs were a good tool in this study, or an effective tool in this study.
Yeah, so we're not, today I'm not going to break you up into breakout rooms, because by the time we get finished going through Gaffney, it's not really going to be enough time to go to a breakout room and then come back and all that jazz. So we're just going to cover everything here together. So can you guys see my presentation?
Yep.
Okay, cool.
It's the PowerPoint view, not this presenter view right now.
Okay, but how is it now?
Still just looks like PowerPoint. We can see your slides on the left.
Well, stinker. All right. Okay.
There you go.
So who can tell me what a chimera is? I know we talked about this in at least one of the groups that I was in the other day.
Isn't it kind of like a hybrid of like two animals blended together?
Yeah, it is like a hybrid of two animals blended together. So the Sphinx would be an example. A griffin would be an example. An eagle head with a lion body. If you're a fan of Harry Potter, a hippogriff would be a example of that. An eagle's head with a horse's body, etc. So what do they mean when they talk about chimeric DNA?
It's like DNA from two different animals put into like one cell.
Yeah, and in this case, they used which two animals?
Wasn't it human DNA and then like the snake DNA?
It was human DNA and snake DNA. But was most of the chimeric DNA human DNA, or was most of the chimeric DNA snake DNA? Did you guys hear my question? Was most of the chimeric DNA snake DNA or human DNA? This is an image of the sodium channel, and the domains are in this particular publication, the domain labels got offset for some reason, but domain 1 is here, domain 2 is here, domain 3 is here, and domain 4 is here. Which domains were human DNA and which domains were snake DNA in this paper?
1 through 3 were human, and then 4 was snake.
Yeah, so they kept most of the sodium channel was human, but it's just this last domain that they made snake. Why would they have done that? Why not just give it all snake DNA? Why not put all snake DNA in these frog eggs?
It relates back to the question we asked last time about what was the goal of this paper. And we talked about a few different levels, and this gets at that. Why just there?
This down here should look familiar to you because it looks kind of like this here, doesn't it? Where are we at in the sodium channel where all these mutations are occurring that lead to different amino acids? Which domain are we in?
is most of the mutations in domain 4?
Yeah, most of the mutations are in domain 4. And so they just wanted to leave the rest of the sodium channel alone because human beings have tetrodotoxin sensitive sodium channels. So they only changed the last part, the domain 4 sequence, to a snake sequence because they were interested in these mutations in domain 4. They weren't really interested in any of these other mutations in domains 1 through 3. And then they splice these, they clone these into frog eggs. And the reason that they clone these into frog eggs is because then the frog would just take that DNA, and instead of making normal frog sodium channels, it would make sodium channels that were specified partly by human DNA and partly by the snake DNA. And they use snake DNA from these different populations. So we have the 4 populations. This is figure 2. Somebody explain to me what's going on in these figures. Start with figure 2A. Yeah, 4 lines. 2 lines are purple, 2 lines are orange. What does the purple and the orange mean? It goes back to this figure. What's purple here?
Purple is the Bear Lake population, which is very resistant.
Yep, and orange is what?
Bitten, which was more resistant.
More resistant, but not as resistant as Willow Creek. Now, why are there 2 purple lines and 2 orange lines?
Is one based like just on the snake's resistance, and then the second line like from the frog's resistance with the snake DNA?
Not the frog, it's the human snake chimeric DNA. So snake DNA is the open squares, and the chimeric DNA is the closed squares. And all they're basically showing you is that the snake DNA, where the sodium channel is from 100% snake DNA, isn't really different from the human snake chimeric DNA. Because then what they're going to be doing is they're going to be dealing with the chimeric DNA throughout the rest of this figure. What's going on in figure B? Somebody other than Katie. Katie's doing the heavy lifting for you guys.
It's showing the resistance between all the different groups.
Yeah, and it shows that Bear Lake is the least resistant. Then what's yellow again? Warrenton. Then Warrenton, then Benton County, and then Willow Creek. As you move from left to right, the tetrodotoxin concentration is getting bigger and bigger. You have to realize that these are 10 to the minus 10th, 10 to the minus 8th, 10 to the minus 6th, 10 to the minus 4th concentrations. These are nanomolar concentrations, so they're below a mole. And so as you go from -10 to -4, you're getting more and more tetrodotoxin. Then what's going, and so what's the y-axis on all of these figures? I should have asked that at the very beginning. Whenever you describe a graph, you should describe what the x-axis is and what the y-axis is. What is the y-axis here? I mean, it tells you what the y-axis is on the figure, but what does it mean? What does the ratio of unblocked to total current mean?
Do you all remember how tetrodotoxin interacts with the sodium channel, just very generally?
Tetrodotoxin blocks the sodium channels, doesn't it? Or is it
Yes, it
the
does.
opposite? Okay.
Mhm. That's correct.
So then the y-axis is like deciding how much the tetrodotoxin blocks the sodium channels.
Yes. So when there's low concentrations of tetrodotoxin, 100% of the current is generated, and then as that current goes down, it's going down because you're blocking more and more of the sodium channels, and so those nerves can't generate a membrane change, a change in the charge of the membrane. And so this is just basically looking at how much current would be there if there was no tetrodotoxin relative to what happens at different concentrations. And what they pay attention to in all cases is what is the concentration at tetrodotoxin that leads to 50% blockage of the sodium channels. And that's what KD is. So one of the questions here is, what is K sub D? K sub D is the concentration of tetrodotoxin at which 50% of the channels are blocked. And so you see that all of these confidence intervals, and these are 95% confidence intervals, they're all at the 50% mark on the percentage of sodium channels blocked. And then the question is, well, what is the average concentration of tetrodotoxin and how much does that vary? And you see that it's not varying very much, and there are definitely differences between the different populations.
Before you get to D on that, I just want to take a moment. If you're having trouble thinking about the current, remember that on either side of a cell membrane, there are different concentrations of various ions, like sodium ions. So when the sodium ions flow through the channel across the membrane, that's what we're talking about when we talk about current. And that's what's shown or going to be discussed here in a moment in figure D. 2D.
Yeah, and figure D looks very much like the current diagrams that were in the video that I posted on Moodle that I asked you to go and look at. So C refers to just a specific mutation. If they just change one mutation in the Willow Creek population, it results in a difference. It's changing the 1561 from an isoleucine to a valine. So it's basically taking the Willow Creek population and taking this mutation here and changing it from isoleucine to valine. And that's the difference that you get in terms of resistance. So they're still way over here on the resistance end of things, highly resistant end, but that single mutation actually makes a pretty big difference. And in these figures, what's going on is these are the actual current diagrams of a non-resistant sodium channel and a resistant sodium channel. The black line is what happens when a depolarization occurs. You get this huge change in the charge of the membrane, but when you give it tetrodotoxin, that change is much lower because all of those sodium channels are blocked. But in the resistant population, if in a resistant sodium channel nerve, you see that the red line and the black line are virtually on top of one another. If you blow it up, you can actually see that there's red and black there. And this is just, once again, just showing you the current that they're talking about here in B. Any questions about this figure?
So they use that 50% ratio as the metric of resistance, but notice they had to calculate what that is by fitting the line to the actual points.
Sure. So this then just shows the relationship between skeletal muscle and these cloned channels to basically indicate that the channels that they've created are tightly correlated to kind of whole muscle performance. And so these clone channels would be viewed as a good surrogate for what's going on in the muscles themselves. And so this is just to say, you know, what we did in these frog eggs also correlates with what we know about you know, whole animal performance. So what I want to do is I want to focus on what these changes actually are, because there's a lot of meat in here that you don't necessarily get unless you're paying attention to things that you probably aren't able to pay attention to yet. So this was a phylogeny based on the sequences of the sodium channels, and you can see that these are all the sodium channels that we're dealing with, and they are all very similar to one another. This was a crude rudimentary figure for the sodium channel that they did a much better job of in a different paper, Feldman et al. But once again, you can see that they're interested in these mutations in domain 4, and in particular, these 4 mutations here, 1, 2, 3, and 4. This one occurs at 1556. This one occurs at 1561. This one occurs at 1568, and this one occurs at 1569. So what I've done in this next set of slides is I've done some homework for you guys. So these are all of those different mutations, and it's showing you what the sensitive strains have and what the resistant strains have. And in many of these cases, the changes aren't huge, but this particular change at 1568 goes from aspartic acid to asparagine. And if you look at all of the amino acids, you see that aspartic acid is polar or uncharged, and you see that asparagine is positively charged. And so we're basically making a change from aspartic acid to asparagine, going from a polar or neutrally charged side group to a positively charged side group. And when you look at tetrodotoxin, so this is actually the structure of tetrodotoxin, one of the things you'll notice about tetrodotoxin is it has all of these hydroxyl groups. Hydroxyl groups all along the bottom of this, and then it's got this positively charged NH2 group over here. What do you know about hydroxyl groups in terms of their charge? This is basic chemistry.
They're negative, right?
The hydroxyl groups, they are not negative. It's a little more complicated than that. What do you know about oxygen and its behavior towards the electrons of other atoms?
It prefers to gain electrons.
Can you speak up?
It prefers to gain electrons.
Yeah, so oxygen is one of the atoms that we refer to as highly electronegative. Electronegative means that it tends to pull electrons away from atoms and pull them towards themselves. So when you have an oxygen bonded to a hydrogen, it's going to pull the electron away from hydrogen, which is going to make oxygen more negative. And what is it going to do to hydrogen?
Hydrogen would be partially positive.
Yeah, so that's exactly the terminology that we use, right? Like these partial negative and partial positive charges are what go into making polar molecules. Anytime you hear somebody talk about a polar molecule, it's a molecule that has a polarity to it, such that one part of the molecule is partially positive and another part of the molecule is partially negative. But there are full charges like in sodium chloride, where sodium becomes fully positive and chloride becomes fully negative. But these partial charges are actually responsible for a lot of intermolecular interactions. So imagine that each of these hydrogens hanging off, 1, 2, 3, 4, 5 of them, but really it's these 4 right here that are the most important ones that are hanging off the bottom of this thing. Imagine that all of those are positively charged. Now, when you have these positively charged hydrogens on tetrodotoxin interacting with a non-charged side group, then you don't have anything really happening in terms of interactions. If it's slightly polar, they might attract one another. But as soon as you change this amino acid to a positively charged amino acid, the positive charges on the amino acid will actually repel tetrodotoxin because of all of those partial positive charges on tetrodotoxin. And so when we think about the binding ability of tetrodotoxin to this part of the sodium channel, if there's a positively charged amino acid right here and tetrodotoxin is coming in here to bind, it's not going to be able to bind as well if there's a positive charge here rather than if there's a negative or a neutral charge here. If there's a negative charge, it's going to bind strongly there, and if it's neutral, it'll just not have a lot of binding strength at all, as opposed to being positive where it will be repelled. So the interesting thing about this is that when we make this change from aspartic acid to asparagine, when we look at the codon table, this is the codon chart that you should remember from back when Dr. Judd went through this with you. This is aspartic acid and this is asparagine. And so we're talking about this change genetically. These two codons code for asparagine, these two code for aspartic acid, and all you've changed really is you've changed an adenine to a guanine. Because adenine and guanine are the same type of amino acid, they have the same shape. This is referred to as a transition mutation, and it's the most common of all single nucleotide polymorphisms, these transition mutations, where you're either changing out a purine for a purine or a pyrimidine for a pyrimidine. And so that's a really easy genetic change to make, in a sense, from the standpoint of the nucleotides, because it's not changing the shape of the DNA when this mutation occurs. And when you then go from a negatively charged or uncharged amino acid to a positively charged amino acid, that greatly reduces the ability of tetrodotoxin to bind. And if tetrodotoxin can't bind, it's not going to block the sodium channel, and you're going to end up with a snake that is very resistant. Is everybody clear with that? You understand what I was saying during all of that? Any questions about that? They mentioned this in Gaffney, in the Gaffney paper, and they refer to these hydroxyls. Let me go and pull it up real quick.
Well, it occurred to me that I don't remember last year if we talked about why having a positively charged amino acid there, while it repels tetrodotoxin, why that would affect the functioning of the sodium channels generally, the molecular basis of the trade-off. I don't know if we explicitly talked about that. What do you guys know about sodium ions? In terms of charge?
Aren't they positively charged?
Yeah, am I correct in my understanding? I just was trying to think of a good question here. That the trade-off arises because not only is the tetrodotoxin repelled, but the permeability to sodium ions is reduced, in part because there's positive charges at the vestibule, at the entrance to the channel.
Yeah, that makes a lot of sense from the standpoint of the molecular basis of the trade-off. By having that positive amino acids in there, it not only repels tetrodotoxin, which is partially positive, but it will also repel anything with a full positive charge as well. And so it simultaneously makes you more resistant, but then hampers the normal functioning of that sodium channel when it's undergoing its normal work, when it's not being challenged by tetrodotoxin.
It still
so if
functions.
You go over to,
yeah, go ahead.
Go ahead.
I was just going to say, if you still consider the overall structure and function of the sodium channel, it still works, but that might be part of the cause of the reduced functionality.
Yeah, it works. It just doesn't work quite as well. So if you go to page 761, The first full paragraph on that page talks about the change in the charge and what this means in terms of the structure of tetrodotoxin. So it takes a pretty careful reading of the paper in order to get yourself to go and look at what the structure of amino acids are, look at what their charges are, et cetera, et cetera. We weren't expecting you to probably read it that closely and go through the extra legwork, which is why we went through the legwork for you. So that basically kind of sums up everything in this paper. Any questions about this paper? One of the things that we're hoping as you read this paper is that you'll get this idea that molecular biology, which we normally think of as one silo of biology, and ecology and evolution, which we often think of as another silo of biology, that those two silos don't have anything to do with one another. And this set of papers actually illustrates that those two silos have a lot to do with one another. In this case, you can't really understand the ability of this evolutionary arms race to proceed without knowing what the mechanism is of tetrodotoxin acting and what the mechanism of resistance is. And also, when you look at this ecological trade-off between resistance and sprint speed, that trade-off makes the most sense when you can bring the molecular biology of the sodium channel to bear on the question. So hopefully you're seeing that these areas, subdisciplines of biology, are not as discrete as maybe you thought they were before. All right, so phenotypic mismatches reveal escape from a predator-prey arms race coevolution. I need to pull up the other set of questions now. So what was their goal when they were looking at this system in this next paper, the final paper? What were they trying to do here? Joseph, help us out.
Oh man. I am not sure.
Okay. Dr. Judd, I don't have the ability to do this for some reason. Why can I not do this?
Are you sharing a screen or pulling something up? I'm just trying to pull things up as well.
Oh yeah, I'm gonna do a little poll if I can.
Oh, nice.
How do I launch a poll? Oh, there it is. Okay, I'm unable to do that for whatever reason. Oh, there we go.
What's going on here?
I can't launch a poll on the fly.
Mm-hmm.
Okay. How many of you actually read the last paper? Okay, then you guys should know what the paper is about. So one of you guys help me out with what they're trying to ask about this paper.
They were like trying to determine if there was coevolution between the snakes and the newts, or if like it was kind of like, I used the word mismatch, but like, I don't know, things were happening that like didn't necessarily allow for coevolution to exist between the two.
Okay, so if these two things are actually co-evolving, what would you expect to be the relationship between tetrodotoxin production in newts and snake resistance? Somebody other than Katie. Which, by the way, you're doing an awesome job today, Katie.
You would expect tetrodotoxin to increase in newts and then the resistance to increase in snakes.
Yeah, and so what you should see is you should see snake resistance and newt toxicity kind of evolving hand in hand, such that if newts are not very toxic, you shouldn't see very resistant snakes. But if newts are very toxic, you should see very resistant snakes. And so this is what the geographic mosaic of coevolution is. These populations will be matched phenotypically to one another if different selective pressures are happening in these different populations, such that in some places you have intermediate resistance and intermediate toxicity, in some places you have low resistance and low toxicity, and in other places you have high resistance and high toxicity.
Recall we talked about the reciprocal nature of coevolution and the lockstep, right? That's the same ideas.
Yeah. So one of the questions in your discussion questions, which I gave you yesterday in the hopes that you would use those to kind of guide you through the reading of the paper, that's what they mean when they refer to reciprocal selection. Each species is exerting selection back on the other. And so reciprocity. Newts exert selection on snakes, snakes evolve, and then they exert selection on the newt, which evolves, which then exerts selection back on the snake. So in this framework, what are they referring to when they talk about evolutionary hot spots and cold spots?
Isn't it when the hot spots are when they're evolving more and the cold spots are when they're not evolving as much?
Yep, that's true. So under what conditions in terms of toxicity and resistance would you expect to be evolutionary hotspots?
Higher toxicity and resistance.
Okay, what about intermediate toxicity and resistance?
Would you expect evolution to be fast or slow in that condition if you got intermediate toxicity and intermediate resistance?
Slow.
Contrast intermediate toxicity and intermediate resistance with extremely high toxicity and low resistance. Under which of those conditions would you expect evolution to be occurring fastest? If you are an extremely toxic newt and you live around a bunch of snakes that aren't resistant at all, is there selection acting on you? Michaela?
No,
Why not?
because the snakes aren't going to eat you.
Snakes aren't capable of eating you. And so in that case, the newt has escaped the evolutionary interaction. If you have snakes and newts that are evenly matched, what's the level of selection acting on both of those? Whether they're evenly matched at the high end or they're evenly matched at the intermediate level, what's the level of selection that's going on with the two species?
I have a question really fast.
Yeah.
So say you were to take a bunch of really toxic newts and put them in with like very low resistant snakes, because those snakes specialize in eating the newts, wouldn't they eat them anyway? So like they would
And then
still
what
be
would happen to them?
can they die from this, or they would just like learn not to eat the newts or something?
Well, they can totally die from it. Yeah, they could totally die from it if they were really not resistant, in which case there's not enough genetic variation in the snake population to withstand those newts. And so those snakes would just go extinct because they would keep trying to eat newts and they would die. Or the other thing that they could do is they could shift to another prey item. But one of the things that we know about this group of snakes is that they tend towards prey specialization. So if there's very little genetic variability for eating other prey, and there's also very little genetic variability for resistance, and their resistance is low, they're kind of trapped. They don't have anywhere to go evolutionarily. And if they find themselves coming into contact with a population of really toxic newts, then they just wouldn't have any recourse. They wouldn't have any sort of weapons at their disposal. They can't go eat something different, and they can't survive the food that they prefer to eat. And they would just go extinct, and the newts would just live happily ever after. Now, that's not what actually happens in this case, is it? Go ahead, Nathan.
I was just going to say that Casey chimed in on the chat for me, and she said that the hot spot is where the reciprocal evolution is going to be the most intense, regardless of toxicity.
Regardless of toxicity, that's what I'm trying to work us around to. The idea is that when you're equally matched, there's a lot of reciprocal selection going on, and that's where evolution is going to be happening the fastest. If you have severe phenotypic mismatches, that means that one individual in the interaction is way better at doing its thing than the other individual, the other species is in the interaction. And as a result, one of them has escaped selection. There's no longer any selection on that. And it doesn't matter whether it's the newt that's really toxic in a population of snakes that are not very resistant, or if it's a snake that's really resistant feeding on a population of newts that are not very toxic. In both of those cases, that mismatch reduces the level of selection acting on the species that has the most elaborated phenotype. And in this case, well, let's just go to this. Let's just work our way through it. So are they now just looking at 3 or 4 populations? No. Casey, tell me about it.
She's having audio. That's why I'm speaking for her.
Oh, okay. Somebody other than Casey. Casey's having difficulties. How many populations do we have here?
Casey says 20 that interacted with the newt.
Okay, it's 28 actually, according to this figure. So we've got 28 different populations. Look at the way they've written their figure caption. In PLOS, they do it a little differently than some other publications do. You see that they have figure 1, and then they set off the figure title from the explanation. Remember in our instructions to you about writing captions, we always tell you there's a figure number, then there's a title, and then there's an explanation. And in this particular publication, they set the title off from the rest of the explanation. What are the color codings about?
It's different measures of resistance and toxicity.
Yeah, so the squares are resistance in the snakes, and the circles are toxicity of the newts. And it's what we call a heat map. It goes from cool colors to hot colors as you move from low toxicity or resistance to high toxicity or resistance. You notice that there's an Idaho population here where there's an introduced population of newts that comes into contact with snakes that presumably have ancestrally not been interacting with newts. This is probably a pet trade release. Strangely enough, rough-skinned newts are given to small children as pets. What was the name of the movie again? Have any of you guys seen the movie Matilda? Michaela, have you seen the movie Matilda, really? Do you remember what they do to the teacher in that movie? Or the principal?
water?
What's that?
Do they put it in her water glass?
They put a rough skin newt in her water glass. And when I saw that movie, I'm like, oh my God, they're trying to kill the principal. So I don't know that you can buy these in pet stores anymore, but when I was in grad school, you could go down to your local pet store and buy a Tarica granulosa and give it to your 8-year-old kid. And the people in the pet store actually had no idea where that newt came from. And then you can't find them in the pet stores, and maybe you can't find them in pet stores anymore because of things like this map. In some cases, the newts are pretty toxic. In other cases, the newts are less toxic. In some cases, the newts are hardly toxic at all. But depending on where it comes from, you're gambling a little bit if you don't know the provenance of your newt as to whether or not you can safely handle it. And I just wonder sometimes how many kids died of getting the wrong pet from the wrong population.
We had one in my middle school science class. I think it might have been 7th grade. And I was tasked with taking care of it over the summer. Never knew that it was one of these until coming here and teaching this course.
So here you can see where phenotypes are matched. They're matched at Benton. They're matched at McGribble. They're matched at Lata. They're matched up here in the British Columbia area. But then here in the San Francisco Bay Area, there's a really serious mismatch. And so what they've done in this next figure is they've basically spatially extrapolated and created this contour map showing areas of high toxicity, showing areas of high resistance, and then plotting the mismatch, the phenotypic mismatch, the difference between resistance of the snake and toxicity of the newt. And you'll notice down here in the San Francisco Bay Area, there's an area of high resistance of snakes, but you don't see a corresponding high area of high toxicity of newts. And so you end up getting this mismatch. In this case, the mismatch is this difference goes from a low difference is red and a high difference is purple. And so you get this severe phenotypic mismatch here in the Bay Area and in the mountains across the Central Valley. So this blank area in the middle is an arid region of California known as the Central Valley. And these are mountain ranges on either side of that dry Central Valley. And so the newts occur in the moist upland areas, but they don't occur in the dry lowland areas. And so for each of these populations, they basically then go and plot, well, what is the relative resistance of the snake relative to the toxicity of the newt? In this figure, what are the gray areas represent? You have this gray area down here. You have a orangish, tannish area up here. What's the gray area representing?
I'm unable to see anything on my screen. I don't know whether everybody else can see it or not.
Oh, I know what happened. I stopped sharing my screen so I could do something else. And now I need to go back and share again. My bad.
Sorry, I was looking at the actual paper.
Oh yeah. So now can you see? Okay, cool. So they took these populations and they just spatially extrapolated to create tetrodotoxin, toxicity, resistance, and then overlaid these. But when you look at predator resistance here in the San Francisco Bay Area where my arrow is, really resistant snakes. But if you look at corresponding toxicity, it's only kind of intermediate toxicity, such that in the Bay Area, there's this big mismatch. So I'm just basically looking at figure 2 from the paper. And then they plotted these on this graph. And these plots essentially refer to the symbols, the colors in the symbols are newt toxicity, not snake resistance. So they're circles because in this thing, circles were newt toxicity, but they're showing you phenotypic matches and phenotypic mismatches, but these mismatches are situations where what is happening relative to snake resistance and newt toxicity.
The snakes are eating the newts like without any disadvantages coming back to them.
Yeah, so this gray area is situations where the snake is way more resistant than the newt is toxic. It's areas where the snakes are basically impervious to the ability of newts to repel them. What would the orange area be, or the tan area be?
Where the newts are more toxic than the snakes are resistant.
So this is where the newts are more toxic than the snakes are resistant. Are there any populations that they've observed up in that tannish area? It's easy to see. Are there any dots in that area? Thank you, Casey, for shaking your head no. Okay. So the only place that there is a phenotypic mismatch are situations where either the snakes are way more resistant than the newts are toxic, or situations where resistance is really low and toxicity is really low. And then all of these here in this middle area between 85% and 15% confidence intervals here, these are all situations where the arms race is presumably proceeding, such that newt toxicity and snake resistance are relatively evenly matched so that you would have reciprocal selection. So these populations down here in the lower left corner that are all coded purple because the newts are not very toxic and also the snakes are not very resistant, these are not in the arms race. Why? Is it because the snakes have evolved so much resistance that the newts can't keep up? Or is there another reason?
The snake's resistance is high, so the newts can't keep up.
Well, the snake's resistant really isn't high. It's really low. And the toxicity of the newt is also really low. Low toxicity, low resistance. So what do you have when you have low toxicity and low resistance? Are they in an arms race?
They're not even in an arms race. That's why they're not included.
Yeah, they haven't even started. They haven't started down that path of escalation yet. These populations up here are moving towards that path of escalation. These are definitely in the throes of escalation. And then what's the situation with these four populations up here? They're also not in the arms race, but they're not in the arms race for a different reason.
Why don't we go ahead? Go right ahead.
Because the predator has developed like a high resistance, but like the prey doesn't even have high toxicity yet. So,
well, they have kind of intermediate levels of toxicity, but what's happened is the snake has developed much higher resistance than the newt has toxicity. So it's escaped the arms race by basically outrunning the prey item in this case. They've evolved faster, they've developed more resistance, and as a result, the newts are no longer keeping up. And so the snakes have escaped the arms race because they're no longer experiencing significant levels of reciprocal selection simply because the newts haven't been able to evolve as quickly as the snake. Go ahead, Nathan, you look like you want to say something.
Oh, no, I was responding to a chat, but I was going to ask people to answer that question in the chat. And that way everyone takes a stab at it and I can watch the chat.
Yeah.
If you want to use that, we can do that.
Yeah, so you have a set of populations that have not yet entered the arms race, a set of populations that are in the throes of an arms race, and then a set of populations that have escaped the arms race, but they've escaped the arms race, not because the newts have escaped the arms race, but because the snakes have escaped the arms race. Now, knowing what you know about the molecular biology of the sodium channel, why is it always the snake that escapes the arms race?
Because their lives are being threatened?
Well, no, because everybody's lives are being threatened.
Is it because like dictation, it's easier to make it happen in like a lot of the population, like since it's kind of a small mutation, like it can be repeated more easily?
Yeah, so it's pretty easy to stumble across this mutation. And when you do stumble across this mutation with a single nucleotide change, you go from low levels of resistance to amazingly high levels of resistance with just that one change from an asparagine to from an aspartic acid to an asparagine. And so you look at these mass-adjusted mouse units at 50% reduction in locomotor ability, and you see that they're all relatively low until you get to these couple of populations, this one here and this one here, that is the shift from aspartic acid to asparagine at amino acid residue 1568. So rather than this being kind of continuous variation, which is kind of what we are used to thinking about, it's more like a dominance effect where some snakes are not very resistant, and then boom, all of a sudden you have a bunch of super resistant snakes. And that is what leads to these snakes here being able to outrun the toxicity of the newts, is because you can achieve a magnitude of difference, or a couple of magnitudes of difference in resistance, with a couple of relatively minor changes to the sodium channel. Why do these populations not have this level of resistance? What do you know about the nature of mutation? Anybody? What do you know about mutation?
Doesn't it like act randomly?
It acts randomly, and so you have to stumble across these mutations. So that's one reason that there are only 4 of these potentially. What's another reason why not everybody has developed super resistant to newts?
Is it because not everyone has to be?
Well, not only does not everyone have to be, but let's say that you're in a population of medium resistance or medium toxicity newts, or even low toxicity newts, and you happen to be born with this mutation that makes you super resistant. What else does it make you? That was Connor, right?
Yes.
What do we know from Brody and Brody 1999?
It makes
if you're
you
super
slower.
resistant, you're also slower. So if you do stumble across this mutation in a population that is sympatric with newts that are not very toxic, you have this defense against toxicity that, quite frankly, you don't really need, and it has the potential to negatively affect you in your everyday life, slithering around the landscape, because it makes you slower and might make you prone to predation, which would then weed that mutation out of the population. So the fact that these mutations come up randomly, if they come up randomly in the wrong place, There's another form of selection that will get rid of those mutations, because the same mutation that makes you really resistant also makes you slower when you're not being challenged by tetrodotoxin. So this combination of, do you actually need that phenotype when that phenotype has a cost? One of the reasons we had you read that cost paper was so that you recognize that you shouldn't expect these snakes down here to develop extreme resistance, because they're in the presence of really low toxicity snakes. And if they do develop really high resistance, it's actually going to negatively affect them through other means of selection, not selection induced by the newt, but selection induced by predators on the snake.
This links back to the adaptive landscape, right? If your newts aren't very toxic, then there's maybe a fitness valley associated with having high resistance.
And this next slide was supposed to animate, and it didn't. It animated in the wrong way. So here we have showing the response curve of snakes, newt toxicity and performance of snakes across the range of things. And in these gray areas where snakes are unaffected by newts, you see that upping the dose at these low concentrations doesn't really affect the snakes hardly at all. But here in the middle, where we have a lot of reciprocal selection going on, upping the dose of tetrodotoxin actually affects performance quite a lot. And then this is a situation where the snakes would have such low resistance that they would be incapacitated by the newts, and we don't see any of the populations in this case. And once again, it's sometimes useful to pay attention to the figure, because they talk about, for these individual plots, they've normalized the value of TTX, because obviously TTX down here is really low, TTX up here would be really high. And so to make these figures standardized, they've just standardized the variation in tetrodotoxin across each panel. But each of these panels refers to a specific population that is, of course, found on this graph. So you could go to the supplementary materials and you could say, oh, well, what's happening in San Mateo? San Mateo looks like a place where you have really resistant snakes and not very toxic newts. That's a situation where you have snakes not being very affected by increases in tetrodotoxin. Same thing happens at Gilroy and Omo Road. These are all the populations that are in this figure that is actually in their paper. Benton, on the other hand, it looks like as newt toxicity goes up, you're at relatively high newt toxicity. You're also at relatively high snake resistance. And during that situation, you have increases in newt toxicity affecting snakes quite a lot, which is why you have reciprocal selection. And when you go back and look at the map, you see that the Benton County population has snakes and newts that are relatively well matched in their phenotype. These are just close-ups. And once again, all four of these populations that are severely mismatched actually turn out to be in the San Francisco area and in the Sierra Nevadas adjacent to that area. And chances are probably that these are populations that have been separated from one another relatively recently, as the climate has dried since the last ice age. And as a result, they were probably connected at one point, which is why you see the snake populations being very similar in these two areas, even though the newts are not as similar. And then this represents that mismatch between these populations. And I think that's it. Questions? Dr. Jed, have I left anything out?
No, I was going to ask if anyone might have a prediction they could make about the patterns on the snakes. You're trying to tie it all together. Patterns on snakes that are highly resistant.
You mean like a geographic pattern?
No, I mean in terms of their coloration pattern, their stripedness versus checkeredness. I wonder if anyone has would make any predictions based on what you've just learned. in highly resistant snakes, or snakes that are in the arms race and eating toxic newts.
So this is a snake that I observed actually at Scott Lake right here. Intermediate resistant snakes with low toxicity newts. Scott Lake up here. It's the only snake that I saw while I was there. But there it is. Anybody want to hazard a guess to Dr. Judd's question? I actually don't know the answer myself.
And I'm not sure that Brody did, but it's sort of a, how could you think forward based on what you've learned so far about the snake's behavior?
Yeah, Brody hasn't looked at this at all. If you're really resistant, what are you speed-wise?
Slow.
So if you're really resistant, you're slow. Do slow snakes benefit more from being striped or from being checkered?
Checkered.
Why?
Wasn't it
You knew
like
the why.
when they like, wasn't it like when they stop or something, like I don't know. I knew it was something with how they like moved and then stopped, and then like how your eyes continued, so you couldn't tell that they stopped.
Yeah, and so checkered is usually a pattern that helps an animal blend in when it's not moving, actually. Whereas stripes oftentimes stand out. And the advantage to a stripe is really only advantageous when you're moving. Is this what you were getting at, Dr. Judd?
Yeah, that's exactly what I was getting at.
Okay.
Yeah.
Yeah. So it hasn't been done yet, but a good next step, if you want to think about this, is looking at these highly resistant populations and saying, oh, well, this is a snake that has quite a lot of polymorphism in it, just like Semnophis organoides does. Do they tend more towards checkeredness in populations where there's high resistance, and more towards striped morph in populations where there's low resistance? That would be a reasonable hypothesis. I don't know if that's the case.
Yeah, but it links what you all have learned about molecular biology now back to the ecology, so you can see how translating between these different kinds of questions and using researchers who are good at different aspects of their aspects of biology leads to sort of more synthetic stories that pull different perspectives together. And that was something that we touched on in our lab discussion yesterday, that some folks like some parts of the research process better and some folks like other. And when you work together, you get a really nice picture, really nice story.
Now, I will say this. So the subspecies of garter snake that lives in the San Francisco area is unusual in that they look like this. So they are polymorphic. There are ones that are more checkeredy, and there are ones that are more stripy. But one of the things about all of them is they are vibrant. So to think about Dr. Judd's question in that way, these are not snakes that are probably going to be relying a lot on crypsis to get by, which then raises a whole nother set of questions for us to think about. Why would these animals be so brightly colored if they're highly resistant, and as a result, they are slow?
Ooh, that's a good question. Yeah.
It may be that they are mimicking some other snake in their environment that is dangerous, or it might be that in the process of eating newts that are toxic, because they can withstand the newt, they may become what themselves?
If you were a bird, what would happen if you ate a snake that had just finished eating a toxic newt?
Probably die.
You might very well die, yes. Or get really sick and learn to not eat those snakes again in the future. As it turns out, that is an area that they have looked at. They have looked at if whether or not snakes that eat toxic newts acquire toxicity themselves. And you can go read that paper if you want. But as with all science, you get some answers to questions, and then those answers lead to a whole set of different sorts of questions.
We're about out of time. Does anyone
Oh my
have
gosh,
any
we're
reflections
overtime.
or questions? No, all right. Yeah.
We're over time. If you have questions about this, email Dr. Judd or myself. The other thing I would like you to do is, I don't know if Dr. Judd has looked at his labs and his lectures for your responses to the idea forms. It doesn't tell us who has responded, but it gives us a percentage of the class, and mine are down in the low 30s. So don't neglect going and filling out the idea forms for both lecture and lab. I think you have until December 22nd, but don't neglect that. If we get into the middle of next week and the percentages are really low, expect to get another email from us.
Yeah.
And as always, if you have questions, contact us. Nathan, anything?
That's it.
All right. Let us know what we can do to help you get ready for finals, which are next Friday. And we will see you then.
That's it's 11 to 1, correct?
11 to 1 on Friday, yeah. What a way to end the week, Keaton.
Hi, I just wonder where I can submit the self-reflection and like the lab reflection and grading for other people.
Just send those to me via email. That's how I've been receiving them from other people.
Okay, and will you have a study guide for final?
What we can do, so you have study guides from the first 3 tests. So what
Yeah.
we'll probably do is just make one for those last material, and then you already have ones for the earlier tests.
Okay, so
Does that make
just
sense?
use. Okay,
Okay, cool. All right,
thank you. Bye.
bye.